The world of intermolecular bonds. Origin and evolution of metabolism. The life was born through chain reactions by intermolecular bonds as dynamic molecular structures with plasticity. The environment that nurtured metabolism. There are systematic thermal motions in liquid water. Clusters of water molecules are existed in liquid water where atoms are linked three-dimensionally by hydrogen bonds. The heat of melting of ice is 1.4 kilocalories per mole shown in Fig. 1. However, the energy of hydrogen bond formation is about 5 kilocalories per mole. It is an evidence of existence of clusters. Bubbles sometimes appear and disappear in a liquid water. The membrane of bubble is organized by interactions between microscopic molecules at the macroscopic boundary where gases and liquids meet. The metabolism was born in the tissues of intermolecular bonds that can be exchanged between atoms. Even if cracks are about to occur in the bubble membrane, the neighboring atoms will repair beginning of the crack, keeping the frame. Violent thermal movement of small bubbles are observed at defrosting ice of carbonated water. Detail movement of bubbles in liquid water can be observed by playing back video taken by digital camera. Here, comparing a bubble with a diameter of 1 micrometer and a water molecule of 3 angstroms, the volume ratio is 3.7 times 10 to the 10 power. The random movement of individual water molecule cannot move the bubble. The movements of the bubbles are driven by movement of clusters around the bubble. Tandem behavior of bubbles caused by helical structure. A model of tandem behavior of bubbles in carbonated water is the coupling due to a structure with a small volume of low energy extending vertically from the two-dimensional plane as shown in Fig. 3. The model of the lattice structure is shown in Fig. 4 in which CO4 unit tetrahedra rotate alternately and shrink during phase transformation from beta quartz to alpha quartz. Dissolved CO2 molecules will induce helical structure of water molecules. CO2 dissolves well in liquid water. How the molecule of CO2 fits with the liquid water. Freezing ice of saturated carbonated water produces bubbles as shown in Fig. 5. The vacant shaft of the helical structure is shown in Fig. 6. It is considered that the linear CO2 molecule will slip in a vacant shaft of the helix structure as shown in Fig. 7. Organized thermal motions in a helical structure of liquid water. A model of the transformation from beta quartz to alpha quartz is that each tetrahedron of the structural unit rotates alternately with the electric axis as the axis of rotation and contracts. So, the rotational thermal vibration is accompanied with expansion and contraction, as shown in Fig. 8. When there is thermal motion that expands and contracts systematically, the movement of molecules proceeds faster than in random thermal motion. Primitive metabolism was realized by quick response of intermolecular bonds to maintain the molecular structure. Thermal motion of floating matters, those are generated by mixing iron fine powder with carbonated water. The membrane generated by mixing iron powder with carbonated water is shown in Fig. 9. The membrane is formed by intermolecular bond that is expressed as equation 1. We can see that water molecules in contact with the floating matters also move in tandem. The bubble generated by mixing carbonated water with fine iron powder disappear when they stop stimulating. The bubble exists in a state where stimuli are applied, however it soon disappears under the condition without stimuli. The molecules and ions linked by intermolecular bonds are possible to exchange the partner of coupling. This indicates that the mechanism of intermolecular bond allows the organization of molecules to be quickly organized and maintained. The primitive metabolism was realized by intermolecular bonding to maintain the organized molecules. Rapid reaction of hydrogen bond of the membrane is observed in coalescence of bubbles. Bubble and bubble can coalescence into a single bubble. The coalescence of bubbles is an instantaneous event, and it is not possible to observe the progress of the bubbles with the frame-by-frame feed of a digital camera. The fact that bubbles coalesce in a short time means the reaction due to intermolecular bond carried out in a short time. Synchronized movement in floating matter is observed at thawing ice of carbonated water containing amino acids. Amino acids are composed of an amino group, a carboxy group, and an alkyl group CnH2n+1, and are compatible with both water and oil. Therefore, When a small amounts of amino acids are added to carbonated water, the thermal motion of the bubbles is linked over a wide area, and the movement of periodic expansion and contraction like respiration can be observed. The coordinated thermal motion between molecules played an important role in early life activities. Main components of cell membrane of creature were produced in the early Earth's upper atmosphere. Hexadecane and octadecane shown in Table 1 are main components of the cell membrane of creature. These long-chain hydrocarbon molecules cannot be produced by biochemical reactions. The hydrogen ions H+ of the solar wind have a speed of 500 km per second, and when they collide with CO2 in the early Earth's atmosphere, hydrocarbon molecules are synthesized. The synthesized little molecules of hydrocarbon remained in the upper layer, and the repulsive action between the hydrogen atoms led to the synthesis of long-chain hydrocarbon molecules. As shown in Table 1, hexadecane and octadecane are hydrophobic molecule in a liquid state on the surface of the Earth, so it is thought that it floated on the surface of water for many years and accumulated to form the cell membrane of living organisms. Evolution from membrane by hydrophobic hydrocarbon molecules to cell membrane. When an amino acid binds to a region activated by the progress of a chain reaction generated in the membrane of a floating hydrocarbon molecule, and the amino acid molecules are peptide bonded, the chain reaction can be memorized in the molecular structure. The mechanism that replicates the enzyme protein realizes the metabolism due to regenerating the chain reaction. Synthesis of helical molecule in double-layer membrane. If there is a double bond in the bond of the long-chain hydrocarbon of the lipid bilayer membrane, it bends, so the lipid bilayer membrane will become a molecule with chirality that has a different twist. In a homochiral bilayer with a hydrophobic hydrocarbon moiety inside, one output side connects to the other input at the center, allowing the same chirality to pass through helical molecules of the same chirality. It is shown in top of Fig 14. However, molecules with a helical structure cannot pass through bilayers with different chirality as shown in Fig 14 below. The heterochiral bilayer synthesizes molecules with a double helical molecules, such as DNA. Metabolism by the replication of enzyme protein. Replication of protein must arrange amino acid units in a predetermined order. The control and to be controlled molecules must exist separately. Amino acids have a chirality similar to the relationship between the right hand and the left hand. The amino acids of the same chirality are peptide bonded. Therefore, as shown in Figs 0.15 and 16, two thread-like molecular sequences with different chirality are synthesized intermittently at a single point where they intersect. Double helical structure of DNA shows how proteins replicate. As shown in Fig 17, the structure of DNA consists of two strands, i.e. a leading strand and a lagging strand. Both the leading strand and the lagging strand synthesize DNA in the from 5 to 3 direction, but the progression of the chained molecular sequence proceeds in the opposite direction and synthesis of amino acid is carried out at the 1 cross point. The two main chains are connected by base pairs by hydrogen bonds. The base pair is adenine A and thymine T, or guanine G and cytosine C. The combination of these three base pairs for each amino acid are termed as a codon and the anticodon. There are moving connecting points between different rotating chiral molecules as shown in connecting gears. Molecules with a helical structure with different chirality do not mingle. Fig 18 shows moving intersection points of helical molecules with different chirality. Synthesis of DNA in the structure of lipid bilayer. A model of the mechanism for synthesizing DNA is shown in Fig 19. Amino acid molecules are the same size and length, even if they are reversed vertically. However, amino acids are not the same when the atomic arrangement is reversed. Therefore, when ribosome was synthesized, the codon of mRNA and the anticodon of tRNA were set, the structure of ribosome that replicates protein. Fig 20 shows a model of protein replication that takes place in ribosomes, which indicates primitive protein replication in the cell membrane. Since protein molecules need to strictly reproduce the sequence of amino acid units, one amino acid molecule is matched frame by frame as if it corresponds one teeth of a gear, and the step is shifted to perform the next match. The matching of amino acid is carried out intermittently as amino acid unit. Summary. Biochemical reactions are carried out rapidly by the systematic motion of molecules in the tissue of molecules. Initial metabolism was carried out by the reactions that produced the molecular tissues quickly. Movements of these linked molecules were observed by the bubbles generated when the ice of carbonated water is thawed. Protein replication is indispensable for the metabolism of living organisms. It is clarified that the reaction to synthesize proteins is also carried out by the systematic movement of molecules in a three-dimensionally connected helical lattice structure in this presentation. Thank you for your attention.